Everyone. First of all, thank you all so much for joining this webinar. This is the first in a series of at least 2 webinars that we'll be doing looking at clinical trials for a specific kind of therapy for PCD. My name is Michelle Mannion, and I am the Executive Director of the PCD Foundation. And Lynn Ernie, who many of you know from the PCD Foundation, will be helping us run this webinar. And in a moment, I will introduce Dr. Doyle to you. But first, a couple of housekeeping things to take care of. Just as a disclaimer, we think it's very important for the PCD patient community to have information about available therapies and trials. And so this is why we are supporting this webinar, but it should not be construed to be an endorsement of any therapy or trial or intervention. And if you have questions about how this might affect your own healthcare or whether or not it's appropriate for you to participate in any clinical trial, please share those with your personal physician. So the first thing I just wanted to kind of talk about, because this is a little bit new to our patient organization, exciting but new, is clinical trial and what is actually meant by that term. We have been involved in studies. We've been involved in clinical studies as a PCD Foundation, looking at natural history and some of those sorts of things. But when we're talking about a clinical trial, what we're really talking about is an intervention that is meant to produce change, hopefully better outcomes. And those kinds of interventions are very highly regulated, and they need to be approved by the FDA. And it can take quite a bit of time to get that done. As you all know, as a patient community, we have some obstacles. We're a very small group. We're a very genetically diverse group. We're also a geographically diverse group. There aren't a lot of people in any one place with PCD. So getting clinical trials done has been very challenging. They're extremely expensive. They require a lot of participation in order to be valid. And it's just been kind of a slow process trying to get us to the point where we are ready for clinical trials. Several years ago, actually more than a decade ago, we introduced what we called the path to clinical trials. And that was a PCD Foundation effort to overcome some of these obstacles by creating a network of clinical centers that could participate in trials and a registry so that we could help identify patients. And those things have both been extremely helpful to getting us to the point where trials are now on the horizon. We still have a lot of work to do. We still have to come together as a community to make sure that this gets done. But we are much farther along than we were 20 years ago when we started. In fact, so much farther along that about 8 years ago, as we were watching the landscape of what was developing in terms of novel therapies that are gene-based or are cell-based, we switched our focus from improving the quality of life for patients with PCD to being focused on getting cures for PCD. So with that background, I just want you to kind of understand how this all fits together for what the foundation is trying to accomplish. We look at interventions from 3 big buckets. And this is just our internal way of doing it. You can look at them many ways, but we think of them as symptom-based. So that would be the things that are treating the symptoms of PCD, antibiotics, mucus thinners, even airway clearance or physical activities that are done to intervene in the disease process. Then there are underlying gene-based kinds of interventions that are focused on correcting the defect, the genetic defect. That is what we will be talking about today. And then the third bucket is regenerative medicine, which we know is also going to be very important in this population because there are people who have significant lung damage and will need something other than the other 2. In reality, probably all of these are going to work together to provide the best outcome for patients, but we're very excited that we have a pipeline developing now with products in all of these different areas. So with that, it is my great pleasure to introduce Dr. Ramona Doyle. Dr. Doyle is an experienced academic clinician scientist. She is an educator and a biotech exec who understands the life cycle of drug development. She has therapeutic development experience with several new and novel treatment approaches, including small molecules, biologics, and cell-based therapies for unmet medical needs. She's passionate about science and about patients and understands what is required for innovative therapeutic product development in diverse patient communities. So with that, take it away, Dr. Doyle.
Thank you so much, Michelle, and to the PCD Foundation for giving me this opportunity, and welcome to all of you. All those things that she read were very nice, but probably of most interest to you and most pertinent is I'm a practicing pulmonologist, and I've been seeing patients for over 30 years, and I continue to see them. And I'm very excited to speak with you today. And I want to focus really on, there'll probably be a lot of questions, but I'm going to get right to it and hopefully leave a lot of time for discussion. So the first slide, please, after this one. Are we? Is our slide move? There we go. Yes, just as a disclosure, I am a paid consultant for Recode. This is an educational session, however, that's designed to just provide you with information about PCD and therapies and how clinical trials work. As Michelle mentioned, importantly, the views that are expressed in this webinar do not reflect the views of the PCD Foundation. And that's my disclosure. So first slide, please. Next slide. So what I'm going to talk about is PCD, and I recognize that many of you may know quite a lot about PCD and others not so much, and I'm going to try to pitch it just right. But if it's too simple for some of you, I apologize in advance. I'm going to talk a little bit also about the potential of mRNA treatments and what it actually is. And importantly, what is a clinical trial? What is it designed to do, and how is it conducted? And then just an update on Recode's clinical development program, which is specifically a medication, a treatment for PCD. And then, like I said, a lot of time for questions, I hope. So next slide, please. So a brief introduction to primary ciliary dyskinesia. Let me have you go to the next slide, please. So let's talk first about primary ciliary dyskinesia. How many people get it? Well, it's a fairly rare disease, but because we haven't really had treatments focused on correction of the underlying defect, I think there's probably a detection bias. That is, we probably don't detect as many cases as there are, and many people are diagnosed quite late in life. So what causes it? So it's basically cilia are little hairs that have many important functions in the body. Hair-like structures that line the respiratory tract, hair-like structures that make sperm motile, hair-like structures that exist in the ears to allow for proper hearing. So what happens in the ciliary dyskinesia is that the underlying proteins that make these cilia are not made to order for the function. And so what happens is the little beating hairs don't function correctly. And that leads to, in the lungs, importantly, an inability to clear secretions as we should, and recurrent infections, and then a lot of symptoms. So the respiratory disease that comes along with PCD is probably the most impactful and the one that's most concerning for really impact on the patients, not only quality of life, but their prognosis. So the symptoms that people have when they have the chronic infections of their lungs that we see with PCD is a cough. They have frequent respiratory infections. They may have noisy breathing, which they'll notice, excess mucus or difficulty clearing mucus from the lungs. And for people whose upper airways, their ears and ear canals are affected, you can get chronic congestion in the sinuses, recurrent ear infections, and cold symptoms. Over time, if untreated, this inability to clear the secretions from the lung and deal with them causes permanent damage to the lungs, which we call bronchiectasis. For male patients where the sperm are affected, they are infertile. And for people who have particular involvement of their inner ear, they have hearing loss and overall a decreased quality of life. So we'll come back to that about the sort of what I would call the cycle. Once these cilia are impaired and don't work anymore in the way that they need to, this whole chain of events happens. Next slide, please. So how is it diagnosed? Well, people with PCD, as I mentioned, often see multiple specialists before they're diagnosed, depending on how they're manifesting. For instance, it's not always the top of mind of someone who, let's say a man seen in a fertility clinic who's infertile. People don't think, oh, maybe they have PCD. It's just not something that people even think about, or even adults with hearing loss. Certainly in the world of respiratory medicine, There's many causes of bronchiectasis, of lung damage like we see in PCD. And the only thing that we really do in a bronchiectasis clinic, if you actually are seen in a clinic or just seen in a general pulmonary practice, is the only genetic testing that's really been done in the past has been for cystic fibrosis. And the only reason we do that now is that we have therapies for cystic fibrosis that are actually related to the underlying genetics. So in the past, also, now that we have the ability to detect some of the genetic foundations of PCD, we've been able to diagnose it now with standards developed by the American Thoracic Society, professional societies of pulmonary doctors. In the past, the only way to really diagnose it was to do a biopsy of some tissue in the nose or somewhere where you could actually look at the hairs and you can see that they have the structural problem because of this lack of the right proteins. So the way we diagnose it as doctors is we take a history of a patient who's had frequent pulmonary infections, cough, physical exam. Often we'll hear these wheezes and whistling or noisy lung sounds. There are some tests that we do. The nasal nitric oxide measurement is a test that sort of tells us how functional are the cilia that are there. In other words, is it high or low? And that tells us a little bit about the underlying function. Also, the microscopy, high speed or transmission electron microscopy, you actually can take a look at from a sample from a patient and see how these hair-like cells are beating or not properly, and that can be part of the diagnosis. And then genetic testing, which I've mentioned. The challenge is, of course, that there are many different genetic versions of PCD, but we'll get to that. So, but it is a frustrating thing for patients because you may go to many physicians and they'll just say, oh, you just have asthma or something else. And so it can take a long time, and patients can have substantial damage to their lungs by the time that they're diagnosed. Next slide. So right now, I've said that there's not really a cure. There's never been a fix for this abnormal beating of these cilia because there just hasn't been. We didn't have the technology to do it. So what do we focus on now? And it's important. We focus on symptom relief. How do you feel? So all these things are incredibly important. What we're talking about will not replace this anytime in the near future. But once you've got this problem, you've got to practice avoiding future problems and complications. So clearing your airways regularly of mucus and special techniques or bronchodilators, that's a regular part of this. If you have a low threshold to treat anything that seems like an infection, because once the lungs are unable to clear themselves properly of mucus, the mucus becomes a focus for other infections. So aggressive and early treatment for infections. I mentioned bronchodilators. For inflammation in the nose, nasal steroids, and also sometimes inhaled steroids in the lungs or other anti-inflammatory medicines. IV antibiotics are required sometimes depending on the severity of the infection, and sometimes depending on the amount of permanent or temporary damage to the lungs in a bad infection, you may need to be on oxygen. And in fact, over time, some patients with very severe lung disease require supplemental oxygen. So I want to talk about mRNA treatments, and this is going to be it's complicated because there's been a lot of misinformation, but I want to try to boil this down. And probably the thing that most of you are familiar with most recently is vaccines. And I'm in the use of mRNA in vaccines, but I'm going to talk specifically about the broad concept of what mRNA treatments are. It means messenger RNA, by the way, which will help you understand it. So next slide, please. So what is mRNA? When cells are talking to one another and taking signals to do this, that, or the other, there's instructions that go back and forth, signals, chemical signals that tell cells what to do. Messenger RNA literally is a message that goes to cells in the body that tells it to make stuff. It says, this protein is like in PCD, where the protein that helps give you normal beating hair-like structures is missing. The mRNA tells the cell, make this protein. And so it's the signal that's lost to tell. So it's just, I call it like, it's an envelope with a message delivered to the cells that says, you need to make this protein. And the mRNA vaccine was a message in an envelope to cells that said, make antibodies to this virus, because it could hurt you if you don't have antibodies. So it's a set of instructions. What it is not is it's not going in to the whole DNA sequence and clipping and cutting and changing things in your DNA. It is simply a way to package a message to the cells to make a protein that's not made in patients, or not made well, or it's only made in a defective way in patients with PCD. So what you do with mRNA, it's a set of instructions, and it's a signal to the cell that says, hey, you need to make this protein, because that protein, the normal protein, is missing. So how do we get it into the lungs of patients? We can actually deliver it directly into the lungs with a nebulizer or an inhaler. Now, interestingly, I talked about the fact that this is just a message that says, make this protein. If you stop giving it, it won't make the protein, the mRNA message. So this medication, an mRNA treatment, actually has to be given regularly, like a tap on the shoulder to the cells that says, hey, you need to keep making this protein. And importantly, what that says is this is not a permanent alteration of the DNA. This is just using a message system to tell your cells to make something. So people often have a lot of misinformation about, well, if we do anything genetic, it'll affect all the genes in my body or all the cells in my body. Well, that's not actually true, and particularly not in the case of mRNA. So it's a complicated topic. And by the way, there are experiments where people are using types of therapies where they do go in and directly change things in the DNA and RNA, but that is not what this mRNA treatment is all about. I'm sure there's going to be a lot of questions about this, and I hope this has started the conversation. It can take many go-rounds to make the connection here, but I'm happy to take questions. So let's carry on and get to that. So that's what mRNA treatment is. Messenger RNA, just remember the messenger part, and that's helpful. Next slide. So now I want to talk about clinical trials, and clinical trials are done in stages, and I'll just try to break it down, and we'll talk about that, and then we'll take questions at the end. So next slide. So what is a clinical trial? A clinical trial is a trial that tries to look at new ways to either prevent a disease, to detect it, like trying to detect breast cancer early, or to actually come up with a treatment for a disease. And why do we do them? We do them, first of all, any drug that's ever going to go into a human being as a treatment has to go through a very elaborate set of studies, mostly designed, first of all, to be sure that the treatment is safe to give to people, and of course, to be sure that it's effective as well. So a participation in a clinical trial is entirely voluntary. So you should remember that. It's never a requirement. So next slide. So why would anybody participate in a clinical trial? Well, first of all, it's the only way that we've ever gotten, we'll ever get new drugs and new treatments and new interventions approved here in the United States and in Europe and across the world. And we learned so much, I mean, in terms of what we could do differently or how we might attack a disease differently, and really whether something works. So part of participating in a clinical trial in a patient is an opportunity to advance the science, the understanding of the disease, and in the course of that, we may or may not come up with a new treatment, but inevitably there's a lot of learning that can be applied. The other is a really altruistic motive. You want to help other people. While you understand that being in a clinical trial may not directly benefit you in that moment, you're laying the groundwork for improvements for patients with the same or similar conditions in the future by participating in new science. Another sort of side effect of a clinical trial is that you do potentially are in line to be on the cutting edge of a new therapy as it sort of comes through the pipeline. And frankly, in this country where not everybody has the opportunity to have all the healthcare that they need, if you're in a clinical trial, because clinical trials require close monitoring of patients, you will actually get an extra set of visits and care from the clinical staff, because that's required for patients that are in a clinical trial. So why are they important? Well, we want everybody wants new medicines and new treatments and new interventions, and we want to learn about their diseases. And this is how we determine whether those new interventions, medicines are safe, first of all, and whether they actually work. And new medicines that are going to be available to patients cannot be approved and put out unless you have good evidence in the form of a clinical trial that they actually are safe and that they work. So who are the people involved in a clinical trial? So just a couple of the players so that you know. I've talked about the FDA. We're fortunate in the United States that we have an agency that oversees clinical trials to ensure that they're designed, conducted, analyzed, and reported to federal law and good clinical practice. The FDA is the ultimate arbiter of the data that's produced in a clinical trial, and they decide whether the drug should be approved and provided to patients. The sponsor is the group or company or organization that's actually initiating, managing, and funding the trial, and that has to submit all of its data to the FDA. Principal investigator will generally be the person responsible for leading and conducting the research, and at your wherever you were taken care of, often that's the physician who's providing the care. An institutional review board is another level of oversight that's usually local to your hospital or clinic or university or wherever you get your care, is an independent committee that reviews and monitors all of the research that involves people. It ensures that the rights of the participants in a trial and their welfare is protected. So it's completely independent from the sponsor, the investigator, and from the FDA. What is a protocol? A protocol is a roadmap. It's a document that describes precisely what we're trying to accomplish in the study, who the right patients are to participate in the study, and what the schedule of visits, treatments, tests, and procedures are going to be, and what the length of the study. So it's kind of the roadmap. If you're thinking about participating, it tells you what exactly you'll be doing in the course of the study over time. So those are some of the key roles and the key terms for people to be aware of when you're thinking about clinical trials. Next slide. So I want to talk about clinical trial phases, because this is a shorthand often that you'll see or people will refer to the phase of a trial. So I want to start first and talk about preclinical. Well, it's exactly, preclinical means before it gets into human beings, okay? The research about a treatment before it's tested in humans can start in a dish. It can proceed into different animal species or different cell-based preparations. But the preclinical package of information has to be submitted to the FDA. And it includes, by the way, important safety studies, like, is this a drug that could hurt humans? Does it have any side effects, et cetera? That information goes to the FDA, who then decides whether or not you have permission to begin a trial in human beings. So the preclinical work, research about a treatment can go on for years before it actually makes it to the clinic. For instance, people often said of the COVID vaccines, oh, they're too new, I'm scared about it. Well, let me tell you, the research on that mRNA vaccine was going on for 20 years before we actually got drugs. We were just very lucky that it happened when we needed it. So there's a significant hurdle in the preclinical to get into human beings. So what is a phase 1 study? Phase 1 is usually are usually small studies that generally start with healthy volunteers who don't have the disease to just learn about the safety and how the body metabolizes the treatment that they're given, and look at potential for safety or side effects, and also sometimes get a feeling for what's the range of doses that would be right in human beings. Some phase 1 studies actually start in people with the disease. That's typical in cancer trials where people are quite ill and they wouldn't want to, and some of the side effects from chemotherapy we know can be severe. So they often start in patients with cancer. But in general, and in the case of PCD and the trials we're going to talk about later, there's already been a trial in healthy volunteers to get a lot of this basic information. So phase 1, is it safe? How do people tolerate it? Phase 2 is a slightly larger group where you now start to look at people with the disease and look for a signal that it's actually doing the thing that you want it to do. Okay. And often that signal will be like the study of the little hair cells. You see them beat more normally, or something in the lung function changes, or so you're starting to look for a signal that there's efficacy and a reason to believe that you should keep going. The phase 3 trials are trials that are required by the FDA to get approval for the drug. And what the design and how it's conducted is often a negotiation between the people doing it, the patients participating, and the FDA to say, what would success look like? What do we need to see to believe that this drug is going to work and be helpful to patients? So phase 3 trials are typically larger. There's more elaborate, there's ongoing safety and efficacy through all these stages. And often the studies will compare different doses against a standard of care. So importantly, if a drug is no better than a drug that's already available, then it's not going to offer much benefit to patients. So the idea is to really show this is a significant improvement. And if the FDA approves it, generally following approval, there is a very extensive and long-term ongoing safety and monitoring, and some studies around, is it cost effective? Has it reduced hospitalizations or the need for oxygen? Is it actually impacting patients in the real world? So that's a shorthand for the phases of a trial. So as you go from animals to smaller healthy volunteers to get very basic information about safety, then into small groups of patients, then into larger groups of patients, and you importantly compare any new therapy against standard of care. So those are the phases of a clinical trial. We'll go to the next slide. Again, want to have plenty of time for questions. So when you're in a clinical trial, or you're interested in a clinical trial, how do you do that? Well, there are many ways to express your interest. Sometimes it's through the folks taking care of you. Sometimes it might be through the PCD, which would probably be a good trusted arbiter of clinical trials. So the first thing is to see, is this particular study, or do you fit the criteria to be in the study? So there's some because you don't want to go through all of this and then find out you're not qualified to be in the study. So the pre-screening is designed to find out, would the study be appropriate in somebody with your particular manifestation of your healthcare problem. So if you've passed through the pre-screening, the next thing is an informed consent. This is very important. This is a document about that you feel that you understand what's involved, and that you are understanding all of that, you still want to participate. And I will just say this about informed consent, but you should feel comfortable signing it. You can always change your mind about being in a study. But this is a way to be sure that your rights are protected and that you're fully informed. So what's this? The first visit would be called a screening visit, where you come in and you have lots of exams and tests. Again, there is some pre-screening, but there may be some very specific things they need to know about you that they won't know unless they get a blood test or lung function test first. So the first visit is, again, to see, do you fit into this criteria for this particular study? And at that point, there's a lot of information gathered. And assuming that that's successful, then you begin the study, and you'll have study visits throughout the study where you are benchmarked. Often it's to study dose and absorption, but just to see how you're doing, be sure you're safe, that you haven't had any side effects, but also to see how you're getting along. And if there's any improvement in any of these parameters. By the way, you may feel improvement, but the results are saved for the FDA analysis. So nobody knows ahead of time. They can't fix the results. It's all the investigators are blinded to the results of the study visit details. So once you've gone through the study visit, you've gone through the study, there is usually a wrap up or a final visit where you come in and you again look at the end of the study and the data. The FDA and the sponsors will begin to look at the data and analyze it. So that's pretty much what your journey would be like. There may be multiple study visits, and depending on the trial, the spacing of the study visits and overall the length of the study can vary. Next slide. So we're going to have another webinar about Recode research, and where things are with Recode, importantly, is many years of work have gone into the preclinical data, and Recode is now, I believe, been granted this IND to actually study the drug in patients. A study has been done already in healthy volunteers, but the next step is to do the study in patients with PCD. So that's where we hope all of this progresses, and there's a lot of, and I don't want to overstate it, but this would be one of the first correct the underlying defect in a subset of PCD patients. I mentioned that there are different genetic versions of this in a subset of PCD patients, but hopefully the future for all of the different genetic variants. And I'm going to, if there are questions about that, well, I know we have some folks from Recode on the line. So next slide. Good. So we're on to the question and answer. That was a lot of information. And I am hopefully whet your appetite for more. So I think we'll just open it up for questions. And I may invite, if there's some things that I'm not as well informed about, I might ask. So I see there's some questions already.
Yes. So how we normally do this, and Lynn, can you just remind the people online, should they go in Q&A or should they go in chat? I can never remember on webinars. Should they put their questions in Q&A?
Q&A. Yeah.
They should only see the Q&A.
Okay. Yeah. And then so I'll field them to you if that's okay.
Yeah.
Just so
yeah. I can see them in front of me.
Okay.
So
one of these things, and we knew this question would come up, it has to do with gene selection. And I wonder if you could address why DNAi1 and maybe how the message and the size of the envelope affect how genes will be selected.
Yeah. Okay. I'm gonna turn that over to, I'm going to turn that over to Dr. John Matthews, who's a pulmonary specialist who is at Recode. John, are you on? I'm not as, don't have the same detailed understanding of their pipipeline.
I don't think he would be able
He's
to.
not able to. Yeah. Well, we'll come back. We'll come back to that.
Can you unmute him, Lynn? Is it possible to do that as a participant?
No, I'll email him. He'll have, I'll have to send him a link.
So we should come back to that, because I think that's an important, and you obviously noticed, Mr. Marcus, that this first trial is in the DNA, another subset, not the H5 mutation. So I would imagine they'd say as fast as we can get the product made, and but we'll wait till they come on board.
I just would like to make a note, comment to the community. There is a lot of preclinical work in DNAi1, so it made sense to go with that gene. Yeah. And what we're hoping for here is that we hit it out of the gate with the first gene, and that will really encourage.
That'll open it up. Yeah.
Yeah. So it wasn't selected for any reason other than it was the one that made the most sense to go with.
Yeah. And there may be details about the preclinical models, and they were just more available. So I hope they'll do all of them very soon. But yeah, thanks.
So this is also this came up from a family the other day, will mRNA therapy work for all the kinds of mutations in PCD? We know it's almost entirely autosomal recessive, but there are dominant strains, there are
Right.
X-linked.
Yeah. Yeah, no, I think that's I mean, the idea is how well do you write the instructions and for which genetic correction? This particular one wouldn't, I don't think, for one genetic, wouldn't be expected to work for all of them, but I would imagine that there is interest in seeing if there, anything other than this one would be developed and at what time. I just don't know. mRNA therapy does have absolutely has the potential to treat all of the genetic mutations. It absolutely does. A very specific set of instructions will have to be given to those cells about what the actual problem is in what's gumming up the work, so to speak.
Yeah, I think that just reflects again on the more success we can have with these early trials, the easier it will be to get funding to continue doing trials.
Yeah, it's a huge hurdle, because if you imagine, and we were talking about this earlier, each subset genetic subgroup is a rare disease, is a version of a rare disease within a rare disease. And the hurdle for showing efficacy, and you've got a small group of patients to start with, can be very high. So one of the things we have to start doing is have this community become aware of, how important participation in clinical trials to create momentum, to say, we want people in this space developing very specific things for each specific mutation.
John, if you want to check your email, I sent you a link so you're able to join.
Yeah, so we can go on down there.
We have a couple of questions related to therapy versus cure, and how often the dosing would probably have to.
Yeah.
If we know that, I don't know if that's known.
Yeah, I think they do have a handle on dosing. I mean, we talked about correcting the method. There are I mean, that's what this particular mRNA therapy is about. Could there be future work underlying correcting the genome at a upstream level? Yes, there could be, but that's not what this particular therapy does. So I see a couple of simple questions like starting phase 2, and actually that phase 1 in patients is the next study. There's been a phase 1 of healthy volunteers, and now the next study would be phase 1. Sometimes a phase 1 can blend into a phase 2 in the sense of, if you start to see signals, you may expand that and start to collect more data. So sometimes those lines are blurred depending on the design of the trial. Yeah, and generally there has to be a control group in every study pretty much, to be able to determine differences in the therapy versus just the difference of being in a trial. Not necessarily in the earliest phase trials, because you're just trying to get basic information, but certainly a safety signal, you have to know whether it was the drug or it was the effect of being in the study. So control groups are used in all of the phases, generally speaking, except in some of the phase 1s.
So I'm assuming we don't probably have all of these answers yet, but worth bringing up.
Yeah,
because
these are great questions.
The delivery is right into the lungs. Would this be expected to help with infertility or the upper airway manifestations of PCD?
Yeah, that's a good question. And I'm still waiting for John to come on, because I don't want to misspeak on behalf of Recode. I think that the idea is the most morbid and difficult complication is in fact the lung disease. And so the idea to get the message and get the protein so you can actually correct and clear secretions. Delivery for the other issues, I mean, the upper airway, the nose, that could certainly be, that's, I think, been contemplated. The inner ear and fertility, those two things, I don't really have an ability to comment. You would think intuitively, though, that might require a systemic or some other type, either a local delivery to that area or some type of systemic therapy in the circulation. It just so happens that the lung the lung is right there, and you can inhale medicines into it and deliver it directly to the lungs.
I love these questions. You can see
They
there's
are
a lot
so
of
great. This is a pretty, I feel
enthusiasm.
like I've insulted this group. They're very sophisticated in their questions.
One question is related to the age of people who would be involved in these, at least initial trials.
Yeah.
Is too young?
That's a good question. Well, the FDA thinks it is. We generally have to prove that something is safe and efficacious in adults before there's a series of steps to be allowed to study things in children. Now, obviously, in a disease that's only in children, there are exceptions, but typically you have to do some work in adults before you're given permission. But importantly, the intention would be in any development program to get the drug to everybody who needs it at all ages. There's just because of this, the regulations, which I think are designed to protect patients. You start with older patients, you may then go to teenagers and then different age groups. Some of you might recall we had COVID vaccine pretty quickly for adults, but the trials that proved it safe and effective in children came later, because that's dictated by making sure that it's safe in adults first. But absolutely, I mean, the pediatric population here, what we want to do is get in early and prevent these problems for developing, but you have to start in adults as per guidelines and safety concerns.
So we have some questions related to how trials would work. First of all, how expensive is it to do a clinical trial, especially this first clinical trial? Would that need to be repeated for every single gene, or would we not have to do?
Wow, that's a really good question. I'm going to talk. I've been in drug development for a while. I don't know exactly how much the Recode trial, but I would say for a phase 2 trial, it can be anywhere from $20 to $50 million. Phase 1.
I think it
depends on the size of it. So the idea is, yes, if we have to do the whole pathway for every trial, for every genetic thing in PCD, yes, it could get. But I think there are ways we can think, and I know Recode scientists and staff are thinking very creatively. Is there a way in negotiating with the FDA that we negotiate smaller trials, maybe with endpoints that are good surrogates for the ultimate endpoint, and to try to speed things along? I mean, we want to get this to patients as soon as we can. So I think that's a great question. And I actually think this is a great dialogue between the PCD Foundation, patients, and Recode, because it sounds like some of you guys have some good ideas about how we could do this. Because it's a hurdle. It is a hurdle. People with rare diseases don't get drugs because it's too expensive and too hard to develop them. So you have to try.
Part of the reason for that, just so the community knows, is that there are not enough patients in any one location to do a valid clinical trial. So it's always got to be multiple centers. A single multi-center trial will cost more than what the PCD Foundation has managed to raise over our entire 20-year existence. They are very, very expensive. Yeah. So that's why it is also important for us to have really good results out of the first time.
Well, we hope. And I just want to say this. I mean, you have to be resilient if you're in drug development. I mean, it took 20 years to get the mRNA vaccine ready for prime time. I know that Recode is committed to remaining in this space. This isn't for the faint of heart, this type of therapy in people with rare diseases and people that, I know that they're very passionate about it. So yes, we would like to see positive results first, but I think that also, I know that they have plans to look at other genetic variants as soon as they can. So
we have some questions on exclusion and inclusion criteria related to how do you know which what your genetic hand is in PCD, and will there be exclusions related to underlying defects like non-tuberculous mycobacterium infections, those sort of things?
Those are great questions. Genetic testing will be part of the study, right? I think so people who haven't been tested will be able to get tested. Typically, in terms of, you want to be as inclusive as you can, but you don't want to put anybody at risk for something if they don't have to be. John can speak to the mycobacterium. I mean, just off the top of my head, because you'd be probably on background antibiotics and there may not be the right window for the study, but it looks like John has joined us. So we're on the inclusion exclusion criteria, but we might want to back up because I know there were a few questions that I didn't answer that people had. So I'll let you two speak.
Hello, everyone. So yeah, thanks for letting me join. I'm John Matthews. I head up clinical development at Recode Therapeutics. And it's great to be able to join and to work with PCD Foundation to have this dialogue as we just embarking on the ability to do our first clinical trial dosing a PCD patient.
Exciting.
There was a press release March of this year when we started our healthy volunteer study with this mRNA for DNAi1. And I think the original questions that came up were really like, why DNAi1? And I think Michelle Mannion and Professor Doyle spoke to that, is that it was the first genetic identification of a genetic underlying cause for PCD. And so there was a lot more known about DNAi1. There were some preclinical models, and the size of the protein, DNAi stands for intermediate. And so it's an intermediate sized protein in this protein machinery that causes the hair cells to move. And so it's a good one for us to start with. We think it's got a higher probability of success, and it gives us proof of principle that we can then change out the mRNA and start to look at the other genetic causes of PCD. We would have a strategy where we would try and get through as many of the genetic causes that allowed us to get to at least covering half of the disease, half of PCD's genetic causes. It becomes much more difficult to develop a drug because as you get to the less common causes, it has what I call a long tail. So say DNAH5, for example, is 20% of PCD, DNAi1 is 10%. So if you add those two together, you've covered 30% of the disease. But there are lots of people in the audience that won't have either DNAi1 or DNAH5. And so it's like, where am I going to be helped? And so the intention is once we start to get proof of principle, we can start to really get the engagement to really cover as many as possible. And I think this is the beginning of a dialogue. I don't want to overpromise. My focus is really on getting the DNAi1 through as quickly as possible so that we can have this proof of principle.
I just want to say again, I love these questions. I love that everybody's really thinking about these things. We have a question on if people do want to enroll in these trials, will they need to stop taking their current medications? And where will they need to go to participate?
So this is a very regulated industry. So on the December the 8th, we've organized a webinar where we can speak to the exact details of the clinical trial, because we had to send it to an institutional review board that Ramona spoke about, the ethics review, to make sure that anything we say, which is facing you as patients, has been reviewed by a group of lay experts and physicians and healthcare professionals who can look at it from the ethical point of view. So I don't want to speak to the details of the trial until we can use the slides and the material that have been agreed meets the level of comprehension that we have to make sure all our informed consent and all our information can be understandable by everybody. And so we're not leaving anybody behind. I would just say generally that we design our trials so that they're accessible for everybody who has PCD. There will be some criteria where if the lung function is too low, and we would perhaps worry that somebody might be too sick or unable to show the safety of the drug in the first time. So there will be some restrictions, but we can go through those on December the 8th. But generally, we wouldn't want you to stop your medication. You would still do your airway clearance. You would still carry on as normal. There's no interactions that we're worried about that would mean that you would have to stop anything that you're taking. But yeah, I think I've probably covered enough of that question at a high level.
We have a couple of questions, just demonstrating that PCD is truly a global community. We've had questions from the UK and questions from China about when they may be able to take part in these trials.
So the UK, we are submitting the equivalent of the IND to the UK, and then future trials will include Europe. So definitely we will be coming to Europe and the UK very soon. China, we don't have a plan at the moment. That would take additional planning and resourcing. So we don't have a Chinese protocol at the moment, but I don't see why we wouldn't have in the future.
We have a question about the realistic timeline for where we're at now before this something might be on the market and available.
I mean, I can give a general answer in that that's a slide perhaps we could go to of the phases.
Yeah.
Generally, the preclinical, on average, it takes 4 years from a preclinical go to filing an IND is on average 4 years.
Mm-hmm.
Phase 1 is generally about a year. We can generally do phase 1s fairly fast because it's a relatively small number of people. Phase 2s are an average of about 2 years.
Mm-hmm.
And then phase 3 is between 7 and 9 years. So the total from IND to approval is on average 15 years. And these numbers come from some very nice studies done summarizing large numbers of trials. It is a long time, an average of 15. The fastest I've seen from IND to approval is 7 years. And that's doing it fast. So you need at least a year for phase 1, a couple of years for phase 2, and then 3 or 4 for phase 3. And sometimes it's shorter. The fastest I've seen is 7, and then with the middle being 15, you've got some that are taking longer than 15.
Some of that time is that it takes a long time to recruit patients, especially in rare disease communities.
Absolutely.
And so the more we can do to facilitate, if there's interest and patients want to be involved, getting these studies recruited fully, that also will help with the time.
One thing that I would like to put out to the community is that one of the numbers that we know is true is that even in trials involving treatments of cancer, on average, there's only 3% of the people who are being treated for cancer actually participate in clinical trials. That number has always shocked me. It's a very, very low number, and there is big efforts to try and increase clinical trial participation. What we need from the PCD community, I hope, is enough engagement such that we have high uptake of clinical trials, because as we're dealing with subsets of a disease, this is a rare condition to start with. And then as we deal with subsets like DNA1, which is only 10% of PCD, if we have to have high engagement in order to be able to get through these clinical trials in the time span that will allow all of us to see the benefit, not only for DNA1, but then the ability to start the other trials. Mm-hmm. And I hope that doesn't come across as being like self-serving or saying that, we have to do this quicker and quicker. But the reality is that trial participation rates have unfortunately been, I think very low. And I think that's something that hopefully by making trials more accessible to patients, by making them have more flexibility in terms of remote visits, that we need to do better as an industry to be able to do these trials more quickly and more effectively.
Yeah, I think just as a segue to that, it doesn't matter what the intervention or the clinical trial is about. If only 3% of the people with DNA1 participate in a trial for PCD therapy, we're not going to get the validity that we need to get approval. And the reality is the only people who can do this are us. Nobody can do PCD studies for us. We have to jump in and do this. And I think a good model for us is the CF Foundation and the CF community, who really embraced and got involved in therapy. And now they have modulator therapies that are changing the lives of people with CF. So just as a general message, not related specifically to this therapy, just keep that in mind, that this is on us as a community to get these things done.
Yeah, I think that's a great reminder. I'm involved with another rare disease patient group, and Michelle and I were talking about how when I started taking care of patients with that rare disease, there were no therapies but transplant. And now, 30 years into my career, there's 14 FDA approved, but it had to start somewhere. And the patient community being engaged was key, because patients really, really participated in the whole process. So it's critical. And I commend Recode. I think taking on rare diseases is not for the faint of heart in terms of drug trials.
We have a couple of questions here. This is a savvy American audience. You're actually going to be able to get access to the therapy if it's approved, which is always a challenge here. But I'll just weigh in on that and say a lot of that's going to depend on patient advocacy and lobbying to make sure that we have access. We all have access to the therapies that we need.
Yes. Sorry. So the question is will patients in the US have access to this therapy? Yeah. I mean, we certainly described the steps in front of us, so the phases we'll have to go through. So we would definitely take it through all the necessary phases, and then provided there's clinically meaningful benefit, we would definitely have to make sure that there was access. And so patient access to drugs involves the insurers being willing to reimburse for it. And so based on clinical trial evidence, a drug being on label and approved, then I would say we would allow, that we would enable access. So that will be key.
All right. Do we have any? I want to make sure I'm not missing anything here. Have I missed any questions? We have a question about how can you volunteer and how can we make it easy for patients to get in touch with Recode?
John.
So we're planning the webinar on the December the 8th. We'll have a very specific email address that you can contact us. And so we will post our trial on clinicaltrials.gov once it's been ethically approved. And so that will allow a direct route of communication so that we can get all the information that you need.
Good advertisement for next week.
Okay. I think we've gotten through most of the questions, and I'm sorry if I missed anything, but we will be repeating this next Friday. So hold your question for next week if we missed it this week.
Yeah, this is a very sophisticated audience. I'm going to have to bone up a little bit more, John. No,
I just want to thank Dr. Doyle and Dr. Matthews so much for joining us and for sharing this information with our community. And Lynn for saving me on the technology side. I cannot do Zoom. So see you all.
Thank you so much.
Yes, be well. Have a good weekend, everyone. Thank you. Bye now.
Bye.